The next speaker is Dr. James Taylor, CEO and co-founder of Precision NanoSystems from Vancouver in Canada, to talk about microfluidic platform for rapid development and scale-up of nanoparticles for genetic medicine.
Great. Well, thank you very much. Yeah, my name's James Taylor. I look forward to telling you more about our platform and the work we're doing ongoing. All right, so the mission of our company is to accelerate the creation of transformative medicine that significantly impacts human well-being. And with that, what we do is we work with drug developers to really help them to accelerate and develop nanomedicine therapeutics, so really enabling the nanomedicine revolution. We work with our users really from the earliest stages of idea generation, and we have through to preclinical and into clinical manufacturing. And we do this with our technology, which I'll talk about, as well as with our expertise in our team to work with groups to help with any formulation development, as well as bringing your products into the clinic. Shown here is our platform, our technology across the drug development paradigm. At the earliest stages of discovery, we have technology for the manufacture of nanomedicines at the smallest volumes. These are in the tens of microliters, really for screening nanomedicine drug concepts. Our benchtop platform really allows for rapid prototype development of nanomedicines. Our Blaze system to enable larger preclinical manufacturing for second animal studies, toxicology, and CNC development. And we have our clinical platform to bring these nanomedicines into the clinic for human use. And additionally, we support all of this work with our teams, our formulation and clinical solution teams that work directly with our users to help them to progress their drugs and to fill any expertise gaps that they may have. So talking about nanomedicines, this conference here, and very familiar to everyone here, some cartoons of certain nanomedicines shown. We've seen the Alnylam LNP RNA shown there in the middle, really the workhorse today for the delivery of nucleic acid-based therapeutics. Liposome-based drugs on just left of center, polymer-based systems, which we also heard of today, just on the right of center, and various other ones as well. And I would say that as we look at the field of nanomedicine, there's no one nanomedicine drug, there's no panacea that does everything. You really work with these drugs, these technologies to solve a wide variety of different problems. And also, these nanomedicines are very sophisticated drug products. So take a 100 nanometer LNP RNA system, that comprises tens to hundreds of thousands of molecules that need to self-assemble to create that structure. What we've done, contributed as our contribution to the field has been the development of the microfluidic continuous flow manufacturing approach to making nanomedicines. And this is taking advantage of the fluid dynamics at small scales, where we get exquisite control over the physics and chemical environment, and coupling that with our very rapid microfluidic mixing technologies that allow us to have very well-controlled rapid mixing, which gives us exquisite control over those self-assembly processes. Now, this is very important, and the importance of microfluidic mixing is it allows us to get the best nanomedicine product we can achieve. And this is shown with this plot, and we have various other data demonstrating this as well. But as we try to achieve what we call the limit size nanoparticles, so these are the smallest, most energetically favorable structure for a given composition, with the purple plot shown here, we're able to achieve that very readily in the microfluidic environment. Once we start using macroscopic channels and the like, relying more on turbulent flow, we don't achieve the same results. And we've demonstrated this in various different ways, but very important principle here. And with that, we've been very fortunate to help progress the field. There's over 100 publications using our technology in the area of mRNA, vaccines, mRNA for genetic diseases, CRISPR, genetic editing, immuno-oncology, and also combination and targeted therapies and the like. And all these different publications would be on our site for anyone to review. But then also, we're looking at the modalities that have been enabled, enabling things like cancer vaccines, enabling mRNA therapeutics, cell therapy, exosomes, and the like. So lots of great applications that have been leveraged on the platform. Now, thinking about this, and you say if you take this into the context of an mRNA therapeutic, these different therapeutic modalities have a variety of different requirements and constraints. So if we're, say, making a drug for a common disease, you can imagine then the manufacturing of that would require very large volumes and large volume manufacturing format. If we look at other, so the first foray of a lot of these drugs going into rare disease and ultra-rare disease, we start to bring down those volumes, have other constraints around the manufacturability, and really going to, say, the personal individualized medicine, say in the area of cancer vaccines, then you have the challenge of having to make one drug per patient. So very interesting challenges with that, and we'll explore some of this. To resolve some of these challenges, we have our clinical solutions team that really works with clients to provide the expertise required to bring your drug into the clinic, the technology, supply chain, quality systems, and the like. Also, we have a CMO network where we work with various contract manufacturing organizations to put all the pieces together to enable you to bring your drug forward. Now, as we map out then, really developing a big emphasis of going from the bench development through into the clinic. They're just showing here the timeline and some of the details of what you need to progress through to achieve that. And I think this is very important to really have this in mind as you're developing your programs. So going from, say, program kickoff to locking down your formulation and your process, and then starting to work into the clean environment to get towards your GMP batch release, and finally your IND filing. There's a lot of steps involved there and a lot of considerations, I think, that are very important for you to take as a user or as a developer to make sure you're progressing, avoiding risk and avoiding pitfalls. Now, with that, and in the context of the microfluidic-based approach, a big question always is, well, how do you get to larger scale volumes? When people think of microfluidics, tend to think of very small volumes, and in this, in terms of our continuous flow manufacturing, that's not actually the case. So, with our reactors that would be, say, in our benchtop equipment, they would operate between 10 and 20 milliliters per minute. To get to larger volumes, we put them in parallel. So you can imagine a 10 times parallelization of a 20 mil per minute reactor would operate at a 200 mil per minute formulation. And I'll talk about also some new technologies where we can do that in a single device and really push the limits of larger scale volume manufacturing. Now, with that principle and translating through the different scales from rapidly going from bench scale into larger scales, shown here across the different equipment, but each one of these systems is using the same microfluidic reactor. So going from the benchtop system, a 10 milliliter formulation, very rapidly translates into a 100 milliliter formulation, which very rapidly translates into a 1 liter formulation. This is very important with these experiments really happening over the course of a couple afternoons. Additionally, the previous slide was showing siRNA LNP. Additionally, we can show the same thing with mRNA, lipid nanoparticles, again, showing that rapid transition from a 7 mil formulation to a 70 mil formulation to a 500 milliliter formulation by using a parallelization scheme very rapidly, very consistent, essentially getting the exact same particle at each scale and each stage, and being able to do this very quickly and with minimal risk. Just lastly, showing the same experiment, but showing some of the cryo-TM data. The morphology of the particles are very, very important, as many of us know, in terms of where the particles go in the body, how efficacious they are, and the like. And again, getting that supreme consistency across the different scales. Now, I'd like to just spend some time on the microfluidic architectures themselves and some of the physics and what we spend a lot of time thinking about at Precision. Our team is made up of many formulation scientists, but also microfluidic and instrumentation engineers like myself. And so really when we first developed the technology a few years back, was focused around this mixing architecture called the staggered herringbone mixer. And this is a microfluidic mixer that allows us to get very rapid, very controlled mixing by putting in tangential flow fluid paths as the fluids go over the mixing architecture. And this system is in our benchtop Blaze and the parallelized schemes that I showed in the previous slides. And more recently, we've been spending a lot of time working on our Dean Vortex bifurcating mixer, which is a slightly different principle of taking advantage of some centripetal forces and the like to really enable that rapid and controlled mixing as well. And this system actually was developed to do some of the lowest volume manufacturing, the tens of microliters manufacturing that happens in our Spark system. But I'll show you how it also enables very large scale manufacturing as well. Now, looking at performance of the different microfluidic architectures is very important. So this looks at a comparable dimensionality, very comparable system between the Dean Vortexing in purple and the herringbone mixer in blue. And you can see the Dean Vortexing mixer is able to achieve that limit size more rapidly. And so we'd say this is a performance increase where you have a larger dynamic range for which you're able to achieve that optimal mixing principle, getting to that limit size. Just some data to demonstrate that the particles being generated across the two different systems are the same, are comparable between the two different types of mixers, both in the sense of particle characteristics like size, PDI, encapsulation efficiency, as well as the plot shown in the bottom right here showing the in vivo efficacy. Now, interestingly, what we found is we're also able to scale the geometry of these mixers, where the 160, 300, and 600 shown in the legend here are different specific geometries within these different, or within the mixers themselves. And what we can do by changing the size of the mixer for the Dean Vortexing mixer is we can achieve the same profile, the same elbowing coming down to the asymptotic limit size, but enable higher throughput manufacturing given through a single mixer. And with the 600 Dean Vortex mixer shown there, really achieving limit size around 50 milliliters per minute and can push out past 100. We have other systems that really operate around the 200 milliliter per minute range as well. And so with that, as we think about scaling manufacturing, we now have multiple options where we can continue to do this scale out, where we can do this parallelization scheme to get to larger scales. Again, taking the 10 times to a 20 milliliter reactor, getting to a 200 milliliter per minute flow rate. We can do scale up, where we can use 1 times a 200 milliliter per minute flow, or we can use both the scale up and the scale out to achieve very large manufacturing ability, say 10 times of that to be a 2 liter per minute formulation. And with that, I'm completed, but thank you very much for your time. I'll take any questions.
Yeah, thank you very much. Questions from the floor, please.
Yes.
Yes, please.
Do you have a way of controlling whether there's any adsorption onto the channels, or making sure that the flow is continuous over a period of time?
Yeah, absolutely. So, we can control if there's any material and channel interaction. We control that with different material properties. We monitor those types of interactions, and then also just the inherent continuous flow has a the flow through the chip is on the order of tens of meters per second. So there's quite a big push through as well.
It seems you focused on nucleic acid today.
Today, yeah.
Yeah. And, well, I have some experience with your system, actually quite a lot, not some. But
good.
One of the problems that I found is that going to, as long as you deal with lipid that you can work at room temperature or slightly above or below, it's fine. If you need to go to high temperature, you start having major problems. Or at least we used to have major problems with the system.
Yeah, maybe to comment on that. So there is heating functionality within the system to allow heating, say, to work with high transition temperature lipids. So we do have lots of groups making liposomal products using high temperature transition temperature lipids. I think with these processes, with these continuous flow processes, a lot of the what you also need to do is to be getting your lipids into solution, into the solvent-based manufacturing of the mixing processes as well, I think is a key thing with temperature.
I think you'd make a right decision to focus on systems that work well.
Yeah, well, we work with, again, in the publications, liposomes, lots of polymer systems as well. And this talk talked about a lot about the lipid RNA systems. Thank you.
On the left there.
Yeah.
You showed very nicely with sRNA
Yeah.
that you had the similar knockdown using the different chip designs. Have you got the similar data with mRNA?
We will be having it if we don't have it today, yeah. Yeah.
And do you think there'll be any differences, or have you?
There's no indication, so yeah, I don't think so. But we'd like to get the data, demonstrate the data.
Thank you.
And at one point, we should say, so we do lots of mRNA work at the small scales in the Spark system, and just confirming it at the larger scales. But we have done those large mRNA manufacturing runs, up to 500 mils, with those systems.
Yeah, one last question.
So I have a quick question, supply and demand. So if one acquires the system, right, other than the raw ingredients, the phospholipids, lipids, etc., these cassettes for running the microfluidics, right, are they on a guaranteed available basis after the acquisition of the system? So, you set up a system, you run large volumes and large scales. At this moment in time, is your sales scalable to the extent that it can sustain an investor into the system?
Yeah, absolutely. So we have, we have a very sophisticated supply chain. Our suppliers of the microfluidic cartridges that we work with can do millions of pieces per year. There's no real concern there, and we've set up all of our supply and quality system to enable, as we go to clinic, working with our clients to ensure they have all the assurances they need, and we can provide you that. Additionally, we'd go into a supply agreement to ensure that as a user, you have the confidence you need to pursue. Absolutely. Great.
Okay, thank you very much.